Okay, this is a review for photosynthesis, and I'm going to be using a worksheet from my website. Now, you should have this. I think you have this already. If you don't have it, I'm going to show you where you can get it, and in which case, just follow along with me taking up the true-false, and then you can just get the worksheet later on and you can try it yourself. Okay, if you do have it, then take it out and go ahead and use it with me. I'm on my website, Zerobio, here, and I'm going to come down to metabolic processes, and I'm going to grab this review for photosynthesis, this Adobe PDF. So that should open that up, and let's see if I can make that any bigger. Okay, I just have to remember to scroll this so that you can see where I'm at. Okay, all right, so let's go ahead and we're going to try and solve this this thing together. Okay, so the first question or first statement says chloroplasts have two lipid bilayers surrounding them. All right, I'm going to use a check mark for correct, and that is true. A chloroplast, just like a mitochondrion, has a single lipid bilayer. Okay, that's a single membrane, but there's two halves to it, and then another one. All right, so there's the inner membrane, and then there's the outer membrane, so that is correct. Number two, photosynthetic pigments are located in thylakoid membranes. That is also correct. Of course, the thylakoids are these little disc-like structures, and that's where the photosynthetic pigments are located. Number three, the stroma is the site of the light-dependent reactions. Now, that is not true. The stroma, which is this region here, is the region where there is the light-independent reactions, not the light-dependent reactions, so that's false. RUBP is located in the thylakoid. Now, RUBP, ribulose bisphosphate, is part of the dark reactions, or the light-independent reactions, which occur in the stroma, not in the thylakoid. So RUBP, that's the thing that accepts the CO2, and then you go around the Calvin or C3 cycle. RUBP is not in the thylakoid, so that's false. Number five, the sun is constantly giving off energy in the form of electromagnetic radiation. Well, that is, of course, true. Visible light has less energy than gamma radiation. That is going to be true. So if you look at the electromagnetic spectrum, all of those rays, visible light, let's say, is in the middle. Gamma rays are way out here. They have high energy and very small wavelength, that lambda thing. Whereas if you go down in this direction, you get decreased energy, but a larger wavelength. Red light has more energy than blue light. Now, here's the thing. If you know the colors of the spectrum, okay, you're going to have red and you're going to have blue. All right, it's actually the other way around. It's blue here and red here. I mean, if you think of the absorption spectrum too that you drew, okay, it was blue there and it was red over there, okay? The blue light actually has a smaller wavelength. Remember blue at, say, 450 nanometers and red at, say, 650 or 7, all right? So blue has a smaller wavelength, therefore higher energy. So this is false. Red light has a longer wavelength than blue light. Well, yes, we've just seen that, so that's going to be true. Number 9, plants utilize the visible light spectrum to perform photosynthesis. Yes, that is true. Number 10, the x-axis of an absorption spectrum is light absorption. Now, I drew a little absorption spectrum here, and the x-axis versus the y, the x-axis is not light absorption, it's wavelength, normally measured in nanometers, so this is false. Of course, light absorption is on the y-axis, so the amount of absorption of each type of color. Number 11, chlorophyll has a long lipid tail called a phytol. And that's basically right. You got the chlorophyll molecule here, you got the head region, the porphyrin, and then you've got the phytol tail. So that's true. ATP and NADPH are produced in the dark reactions. That's not true. They are produced in light reactions, the light-dependent reactions in the thylakoid. You need light. You make these as our main products, ATP and NADPH. And don't forget, we also make oxygen as a byproduct, and they're created in the light reactions. Now, I'm going to clear all this, but when I clear it, it's going to get rid of, whoops, all of that. And that was false, by the way. Here, let me clear that. That was false. Okay, so hopefully you got all these. If you didn't, just pause the video, go back, and go from there. A stack of thylakoids is called a granum. Yes, that's right. When you have a bunch of thylakoids on top of each other, that's correct. Number 14, a photon is a packet of light energy. Yes. All right, we're not going to get any deeper into the physics than that. Number 15, pigments can absorb light due to their double bonds. Yes, that's the main feature of the photosynthetic pigments, is their double bond system that allows them to absorb light energy. Number 16, our eye see leaves as green because chlorophyll strongly absorbs green wavelengths. Well, this is wrong. If chlorophyll absorbs a wavelength, it uses that wavelength, like it absorbs red and blue. It reflects or transmits the green that comes back to our eye, and therefore we see the green. If it absorbed the green, we wouldn't see the green. It'd be gone, right? So this is wrong. Carotenoids reflect or transmit red light. Yes, that's true. It's the red light that comes out that we see, so they're not particularly using that red light. The y-axis of the absorption spectrum is wavelength. That's not true. That one should be sort of connected to the other one that we did. Let me clear this, and I want to move this up a little bit so you can see it better. Just like that. Okay, so that was the y-axis. That was wrong. A 450 nanometer light would appear blue. That's correct, roughly in that area. 20. The presence of pigments that absorb wavelengths different from chlorophyll allows photosynthesis to occur at most wavelengths of visible light. Now that's true. Although chlorophyll absorbs red and blue, predominantly there are other pigments that absorb all the rest. Kind of like my song, right? Jingle Bells Photosynthesis. True. 21. Chlorophyll B has an aldehyde group, whereas chlorophyll A has a methyl group. Oh, I actually forget. I don't remember. I honestly don't remember, and I'm not going to ask you guys to remember. It's either that or it's the other way around. I don't know. Look it up. Let me know. Number 22. Chlorophyll molecules have a complicated ring structure called a porphyrin. Yes, that's true. That's that head part. 23. Thylakoids are filled with stroma fluid. That's craziness, right? If you've got a thylakoid like that, right, and they've got a little interior, well, the stroma's out there, all right? So that's not true. Carbon dioxide is a reactant in the light-dependent reactions. That's also not true. Carbon dioxide is a reactant in the light-independent reactions, in the Calvin cycle or Dark cycle. The light-dependent reactions are powered by ATP and NADPH. Now, you've got to be careful how you read these things. ATP is produced. NADPH is produced. But those light reactions aren't powered by that stuff. All right? They're powered by sunlight, and they produce this stuff. So this is just not true the way it's stated. Glucose, or a sugar phosphate, is a product, of the dark reactions, and that's true, right? In that Calvin cycle, as you come down here and it goes around, and you churn one off, you can eventually make a glucose or a glucose phosphate there as a product. So that's true. 27. Oxygen is produced by the splitting of water in the stroma. Well, you have to know your anatomy, all right? It's in the thylakoid space that the water is split. It's not out in the stroma. The light comes down, hits the pigments, and it's within the thylakoid space that the water is split. You generate that hydrogen, concentrated hydrogens would then go out through the ATPase, and create the ATP like that. So this is wrong. Photolysis is necessary to replenish lost electrons from photosystem 2, and that's correct, because you've got photosystem 2, you got photosystem 1. The sunlight hits, the electron goes up and it goes down in ETS, and it regenerates the ones in P1, which go over to NADP+. So the photolysis, which is here, splits the water and gives the electrons into photosystem 2. So that's correct. Okay. A photosystem consists of an LHC and an ETS. Oh boy, I wish I didn't have this one. There's been some debate about that, obviously. The light harvesting complex, those antenna pigments, and the trapped chlorophyll. And then you've got an associated electron transport system. When I created this, I think it was a time when we considered both of these to be part of the photosystem. But I think it's appropriate to consider the photosystem being all the pigments associated with absorbing of the light, and the electron transfer system to be a separate thing. Don't get hung up about that. That's just a term. And I'm going to go with correct for now. And we'll say photosystem 2 has the light harvesting complex and the ETS. Photosystem 1, okay, like the photosystem, everything, has the light harvesting complex and the ETS of photosystem 1. Okay, so those things together. I want to clear this again and make a bit more room and possibly move it up. Okay. 30. The light harvesting complex consists of many chlorophyll A and a single chlorophyll B. Well, this is backwards, right? Should be many chlorophyll B and a single chlorophyll A. So this is wrong. Carotenoids and phycobilins are considered antenna pigments. Yes, they are. They absorb sunlight energy, pass that energy eventually to the trapped chlorophyll or the chlorophyll A. So that's true. Light energy is really required in photosynthesis to boost protons to higher energy state. Well, this is not true because protons are H+, and what really gets boosted are the electrons. Remember, light hits it, the electron goes up, and then it goes down in ETS, that kind of thing. So this is false. 33, the reaction center chlorophyll loses an electron to its adjacent ETS. Yes, this is true. Now, whether or not it's an electron or a couple of electrons, we're not going to worry about that. The ETS is embedded in the thylakoid membrane. Now, we're talking about chloroplast ETS, right? Not mitochondrial ETS. So that is true. Number 35. Redox reactions, oxidation reduction reactions, occur as electrons move through the ETS. That's true, right? As the electron goes to an electron component or an ETS component, it becomes reduced. The other one is oxidized as it loses, so this is true. Non-cyclic photophosphorylation does not lead to NADPH production. Okay, we got to be careful. We got non and we got not. Non-cyclic is when the electron goes up, down to the next photosystem, up and out of the system. That's non-cyclic. It does lead to NADPH production. NADPH is there. Remember, NADP plus is the final electron acceptor converted to NADPH, so it definitely leads to NAD production, so this is wrong. Cyclic photophosphorylation does not lead to oxygen production, and that one is going to be true. Because when it's cyclic, we're dealing with photosystem 1, the electron goes up, goes back to the ETS of photosystem 2 here, and it goes down. You don't split the water over here, so it doesn't lead to oxygen production. So that's true. I'm going to clear this up again. Okay, 38. ATP is actually produced in the stroma. That's true. And that's because if this is the thylakoid here, you know, all of that stuff happens on the inside. You have the hydrogen gradient goes through the ATPase, and that's where the ATP is made. So this is the thylakoid right here, and this is the stroma. So the ATP is actually made in the stroma so that it can go over to and be used in the light-independent reactions. Hydrogen ions are concentrated in the thylakoid interior. Well, that makes sense. So that's yes. The final electron acceptor in the light reactions is NADP+, and that is true. Okay, it accepts the electrons, and it's part of the light reactions, and it makes NADPH. The C3 cycle involves fixing oxygen onto RUBP. Well, let's hope not. The fixing of oxygen onto RUBP is the first step of photorespiration, which is a wasteful process. And if you're doing that, you are not creating a 3-carbon molecule that's part of the C3 cycle, so this is false. The C3 cycle is named for the first stable 3-carbon molecule, PGAL. Now, this is phosphoglyceraldehyde. That's the old abbreviation for it. This is not right. It should be PGA is the first molecule of the C3 cycle. So this is false. RUBP oxygenase is the enzyme responsible for carbon fixation. And that's not true. It's RUBP carboxylase or RUBP carboxylase / oxygenase, but it's the carboxylase function that grabs the carbon dioxide molecule, which is a carboxyl essentially, or you could call the enzyme Rubisco. It's not RUBP oxygenase though that's going to fix carbon. All right, so this is not true. All right, I hope that makes sense, right? I mean, if it's really RUBP oxygenase, it's going to fix oxygen onto RUBP and do that photorespiration. So it's not going to fix carbon. One sugar phosphate is formed from two PGALs. Well, yes, in the cycle that goes like this and goes around, when you click one off, all right, you're going to need two of those molecules. Two are going to have to come off, and then together you're going to get a sugar phosphate. Because remember, each PGAL, phosphoglyceraldehyde, has 3 carbons. And to make a sugar phosphate, you need 6. So you need 2 of them. And the remaining PGLs regenerate oxaloacetate. Well, that's just saying like a couple come off here, or one, and then you do the cycle, and then one again. That means as you go back up there, they're creating oxaloacetate. Well, that's in Krebs cycle. I know it's hard to keep this all straight. What's really happening is they're going to regenerate RUBP, the acceptor molecule. All right, so this is going to be false. Now I'm going to move this again. Okay, we don't have that many more to go. 46. Light or low CO2 stimulates active transport of sodium into guard cells, and water follows passively. Okay. We didn't talk a lot about the effect of light on guard cells and stomatal opening or closing, or CO2. We did, however, talk about the pumping of hydrogen ions and potassium ions, the movement of potassium ions, and water. And so what you generally have is, there's the guard cell there. I'll draw another guard cell. Remember, what we had is hydrogen ions pumped out using ATP, and potassium ions flowing in by facilitated diffusion down an electrochemical gradient, and then water flowing in passively following that stuff. All right, so you're going to have to check your notes out. We've covered this in your notes. So we are not talking about the active transport of sodium. All right, so we're going to go with false for that. Number 47, more stomata are located on the bottom layer of the leaf than in the top layer. Now, I'm not being specific about the names. This would be lower epidermis and upper epidermis. This is true, and this is so that when the stomates open, they're protected from the sunlight, sheltered from the sunlight, so that water is not transpired out of the leaf as readily as it would be if they were in the top. Okay? 48. The palisade layer has more spaces than the spongy layer. That's not true. The spongy layer has the air spaces. Of course, you're down here in the leaf, you got the stomate right there, and then you've got cells, you know, here and there and everywhere. But the palisade layer has them all packed together, so when the CO2 comes in, it has a place to go, and then it diffuses into the cells. So this is false. The C4 pathway is an adaptation to cold, wet climates. That's not true. It's an adaptation to hot, dry climates, because you got to close the stomates, which is going to reduce the amount of CO2 coming into the plant. So therefore, that wimpy enzyme, RUBP carboxylase, gets confused. So the C4 pathway is an adaptation to hot climates where you have to close the stomates, and then you're going to have a different enzyme, which we're about to talk about, PEP carboxylase. PEP carboxylase fixes CO2 onto RUBP. Well, this is our normal C3 reaction, where CO2 goes onto RUBP, and then we go around the cycle. This enzyme is not involved with this. PEP carboxylase fixes CO2 onto PEP, which makes sense. PEP carboxylase, add a carboxyl group, CO2 essentially, onto PEP. So this is false. 51. PEP carboxylase can fix CO2 efficiently, even in high oxygen, low CO2 environments. And that's true. All right, and that was the whole point of it. If you have a lot of oxygen, RUBP carboxylase, the wimpy enzyme, gets confused and starts to fix the oxygen. And if you have low CO2, but PEP carboxylase is that adaptation in the C4 plant, it'll take the CO2 in small amounts, and it'll click it onto PEP, and it'll make a 4-carbon molecule, oxaloacetate, which will eventually go and release the CO2. Carbon dioxide is fixed twice in C4 plants, and that's true because the first time PEP carboxylase grabs the CO2, as I just explained, we make oxaloacetate, which goes down to the bundle sheath cells, releases the CO2, which then is picked up by RUBP, and clicked on to our RUBP carboxylase, and clicked on to RUBP. So we do fix the CO2 twice. Once it clicks onto PEP, the other time it clicks onto RUBP. So that's true. Low oxygen and high CO2 tend to increase photorespiration. Well, that doesn't make sense. If you have high CO2, you're going to get your regular C3 cycle, all right? Photorespiration is in low CO2 but high oxygen. Brilliant fall leaf colors occur as chlorophyll breaks down and other pigments show through. This is a little bit of a generalization, but it's basically true, all right? As you look at those brilliant fall leaf colors, you know, chlorophyll is declining, you know, because if you have a decreased temperature, decreased sunlight, those are the two major factors, all right? And then the other pigments show through. Now, there can be some assembling of other pigments preferentially in the fall, and there's some other changes with the acidity of the vacuole and other pigments showing through. But in generalities, the decrease of temperature, decreased sunlight, chlorophyll decomposition, and reduction in production of chlorophyll, and therefore other pigments show through. That's not a bad generalization. During photorespiration, RUBP is broken down and lots of glucose is produced. Okay, that doesn't make sense. Okay, we're not going to produce a lot of glucose because that's a product from photosynthesis that we want, and photorespiration is not giving us that carbohydrate production. 56. Sugars in a plant travel through the xylem, water travels through the phloem. Okay, I want to talk about that. I want to clear this. We are looking at the vascular bundle. So let me move this up a little bit more so I've got some room down there and I won't cut myself off. Oops. Let me see. Make sure you can see that still. Number 56 is where we're at. You know, you've looked at the leaf. This is the leaf. This is the top surface or upper epidermis. This is the bottom surface. And, you know, you look through the section on the microscope slide and you see all these things. And this is the vascular bundle, all right? Bundle sheath cells form the boundary here. This is where that, you know, we're talking about the C4 cycle. Actually, the C3 cycle. The C4 cycle would occur out here into the bundle sheath cells. But I want to talk about these things in the middle. These little tubes that are running through the vein. I mean, if the leaf is looking like this, there's a leaf blowing in the wind, right? You're looking at it. You're looking at this, all right? And you see a vein, so just imagine like the veins in my hand go like this, right? So you're just looking end on, you're seeing a little circle, but it's really a long vessel. There are different types of these vessels, just as there are arteries and veins in your body. There are vessels which are called xylem, and there are vessels that are called phloem. And the way I like to think of it and like to remember is xylem generally carries water and stuff dissolved in water. I should have probably written the word water because W is very close to X in the alphabet, right? So think of xylem as like veins, let's say. They carry water throughout the whole plant. These are the blood vessels, essentially, the vascular tubes of the plant. The other types of vessels are called phloem, and they carry food, which sounds like an F sound. And that's the sugar. So you have different types of vessels within one vascular bundle, as if you have a wire, right? If I looked at the cord of this microphone here, there would be a few different wires in that one bundled cord, all right? And same thing with a vascular bundle. Within this, some of this is xylem tubes carrying water. Some of them are phloem. They just happen to go side by side. All right. So sugars in a plant travel through the xylem. Water travels through the phloem. This is backwards then. So this is wrong. I'll stick with blue. Carbon dioxide concentration limits the rate of the light reactions more than the rate of the dark reactions. What do you think about that? This comes from the factors affecting photosynthesis, and there's very little review of that here, so you will have to know that. But this is not true. Carbon dioxide is involved in the light-independent or dark reactions, right? The light reactions require sunlight. That's going to be a limiting factor. And they produce ATP and NADPH. So carbon dioxide really limits the dark reactions. And you remember we had this kind of curve, you know, where it went like photosynthesis goes up and then it goes like this, right? And if this is light intensity As it goes up and you produce your ATP and an ADPH here, at some point, you can't use it fast enough if you don't have enough CO2. But that's needed in the dark reactions, not the light. So carbon dioxide doesn't limit the rate of the light reactions. It certainly limits the rate of the dark reactions if you don't have enough. And even if you have too much, at some point you just can't use it any faster and it'll plateau. False. This is not going to be true for our test. We'll talk about more plant physiology scientists down the road, but we're not going to deal with a lot of them right now. Basically, the ones we've looked at are Calvin and Engelman. Sugar phosphates may be converted into sucrose, cellulose, or glycogen. What do you think about that? Well, sucrose is glucose and fructose, and plants can make that. Plants can make cellulose. They can also make another polysaccharide, starch, but they don't make glycogen. Glycogen is an animal product. When we eat the starch from plants, we break it into the glucoses and we polymerize it into our own polysaccharide that we like, which is glycogen, which is stored in liver and muscle. So plants aren't making, using sugar molecules making glycogen. And don't let it bother you if it says sugar phosphates. It just means, remember glycolysis, glucose, glucose 6-phosphate? The first step is to phosphorylate the thing. So sugars are often phosphorylated, and the phosphate group helps keep the sugar in a cell. All right, because phosphate is a highly charged molecule, and that repels it from the lipid bilayer. So it's a way of like charging it and locking it into the cell. So anyways, this is false. And I hope that you think plants are cooler. All right. Photosynthesis is a tough sell for a teacher. And I congratulate each and every one of you for your motivation and your enthusiasm for the plant section. And plants are not intuitively, for all of us, fascinating things, but they are incredibly important on the planet, as I'm sure you know. And it's vital that we study this stuff and more. But I hope that this has given you a framework, the first steps of looking at a plant that you would have looked at before and thought, oh, how beautiful, or what a wonderful smell, or eating it, what a wonderful taste. But now appreciating the incredibly complex physiology of these things. And of course, there are a lot of great scientists that make a very good living studying plants and the physiology of plants and how plants can be used for medicines to save lives. How plants can be used as food sources. How plants can be used as building materials, as certain products, rubber, hemp, all of this kind of stuff. All right. So that's the end of my little true false review. Thank you very much for watching.